Junk DNA, unexpected greatness is where you least expect it. Transposable elements, TEs, make up almost half of our human genomes, with 8% being derived from endogenous retroviruses, ERVs, which include long terminal repeats, LTRs. LTRs are often found in open chromatin and potentially act as regulatory elements. But what are regulatory elements? They are gene sequences that can control gene expression. For a simple example, let's say this is the gene for black hair. RNA polymerase is an enzyme that transcribes DNA into RNA and is able to bind onto the promoter for the black hair gene because of a transcription factor. A promoter is a sequence that initiates transcription. A transcription factor are proteins that bind to help RNA polymerase recognize and transcribe the gene. In this case, this works perfectly fine. But when there's a regulatory element seen here, which will be a silencer for our model, actually binds onto the promoter and prevents the transcription factor to work. ERVs originate from infections where the viral fragments interact with host transcription factors via LTR sequences and is then spread by copy and paste mechanism throughout the genome. These TEs were once thought to serve no function. Recently, some have been found to act like regulatory switches. A new study examined LTR subfamilies MER11ABC, which revealed 4 more new subfamilies seen on the side here, to discover their evolution in primate lineages. Lentiviral massively parallel reporter assay, which was used in the study, is a technique used to test the ability of MER11 subfamily in controlling gene expression. Essentially, cells were inserted with DNA sequences and then measured for gene activity. So, what happened? MER11G4 had a strong ability to activate gene expression. It had distinct regulatory motifs for transcription factors to bind to. Further analysis in humans, chimpanzees, and monkeys revealed minor changes in MER11G2 over time. In humans and chimpanzees, some sequences actually gain mutations that could increase their regulatory potential. The study highlights how quote-unquote junk DNA can evolve into regulatory elements that have significant biological roles. Tracing the evolution of these sequences may lead us closer into understanding the role of integrated ancient viral DNA. Transposable elements may have important roles in genome evolution, and their significance may be uncovered through further research. Some strengths of the study was that it consolidated many different techniques for analysis, such as LTR consensus, sequence similarity and network analysis, LTR epigenetic analysis, LTR phylogenetic analysis, and many, many more. Some limitations were that the current annotation of LTR subfamilies is not ideal, as the subfamily division is always changing and can be placed into different subfamilies, posing an issue especially when comparing different species. TE variations can be species specific depending on transcription factor variations and TE landing sites within a genome. I selected this article as it introduced me into the evolution of transposable elements. My Gen 2052 understanding of transposable elements was applied to gain greater insight. I can explore more roles that transposable elements play other than regulation. By reflecting on my knowledge, I gained more appreciation for the otherwise disregarded junk DNA. In conclusion, this article enhanced my knowledge of transposable elements that I learned from Gen 2052. I encountered a new method of analysis and technique that emphasizes scientific application as a means to discover the world and its biological history. I'm intrigued by the endless possibilities of transposable elements that may be unknown as of current literature. Although many parts of the human genome and their function remain unknown, by understanding the genome's many segments, even small ones, will eventually lead to increased knowledge of its functions.